Chapter 4, Microbiology for Sterile Processing Technicians. Microbiology is defined as the scientific study of microorganisms which are living things far too small to be seen with the naked eye. For the sterile processing technician, these invisible organisms are not just abstract biological concepts but are considered the primary enemy of the healthcare environment. Our entire mission in the decontamination and sterilization process is to control and destroy these pathogens to prevent the spread of infection throughout the hospital. To communicate effectively about these invisible threats, scientists and healthcare professionals use a standardized two-part naming system known as binomial nomenclature. This system provides every microorganism with a unique clinical identity composed of the genus and the species. The first name or genus is always capitalized and the second name or species is always written in lowercase letters. In the sterile processing department, you will often see these names abbreviated such as S. aureus for Staphylococcus aureus. Correctly identifying these names is essential for interpreting professional instructions and following safety data sheets, SDS, without error. Because microorganisms are far too small to be measured in traditional inches or millimeters, they are measured using a unit called the micron or micrometer. To visualize the microscopic scale of our work, we must understand that a single micron is equal to approximately 1/25,000th of an inch. To put this in perspective, a single human hair is roughly 50 to 100 microns wide, meaning a micron is nearly invisible even under basic magnification. Most pathogenic bacteria measure approximately 1 to 2 microns in length, which is a critical technical fact for our profession. This specific measurement dictates the engineering requirements for the sterilization wrappers and high-efficiency filters used in the hospital. We must imagine the sterilization wrapper as a high-tech microscopic shield with pores that are designed to block anything larger than 1 micron. If the integrity of this barrier is compromised, the invisible enemy can easily penetrate the package and contaminate the sterile instruments inside. By understanding these measurements, we recognize why even a microscopic tear in a wrapper renders an entire tray unsafe for patient use. While understanding the microscopic scale is the first step, we must now focus on the physical classification of these organisms because their shape and arrangement directly impact how we identify and destroy them in the decontamination area. Bacteria are primarily classified into 3 fundamental shapes known as cocci, bacilli, and spirilla. We must visualize cocci as tiny spherical or round cells that resemble miniature balls under the microscope. These round bacteria often organize themselves into specific patterns that serve as a critical diagnostic tool for clinicians. For example, diplococci appear in pairs while streptococci form long continuous chains similar to a string of beads. A particularly significant arrangement for sterile processing is the staphylococci, which organize into irregular clusters that look like a bunch of grapes. Within these spherical arrangements, we encounter some of the most dangerous pathogens in the modern healthcare environment known as multidrug-resistant organisms or MDROs. A prime example is methicillin-resistant Staphylococcus aureus, commonly known as MRSA. This organism is a member of the Staphylococci family and is notorious for its ability to resist common antibiotics, making it a major threat to surgical patients. Another critical MDRO is vancomycin-resistant Enterococcus or VRE, which is often found in the digestive and urinary tracts. These organisms are highly resilient on environmental surfaces and can survive for long periods on contaminated surgical instruments if mechanical cleaning is not performed with total precision. As technicians, we must treat every instrument set as if it harbors these resistant cocci to prevent the silent spread of infection through our facility. The second primary shape is the bacilli, which are rod-shaped microorganisms that look like tiny cylinders or grains of rice. These bacilli can vary significantly in length and thickness, and some species are capable of forming protective spores, which makes them exceptionally difficult to kill. We must visualize these rods as sturdy biological structures that can easily hide in the serrations and box locks of surgical instruments. Common examples of bacilli include Escherichia coli and the bacteria responsible for tuberculosis. Because of their structural complexity, bacilli require rigorous mechanical action during the cleaning process to ensure they are physically removed from medical devices. The third shape is the spirilla, which are spiral or corkscrew-shaped bacteria that exhibit a twisting morphology. These organisms move with a characteristic rotating motion that allows them to navigate through fluids effectively. We must picture a spirillum as a tiny spring or a coiled wire that is highly flexible and mobile. While spirilla are less commonly linked to surgical site infections compared to cocci and bacilli, they still represent a potential biological hazard that requires effective decontamination. By recognizing these three fundamental shapes and the specific threats posed by resistant organisms like MRSA and VRE, technicians can better understand the biological diversity of the pathogens they fight every day. This knowledge is the technical foundation for selecting the correct disinfection and sterilization methods for every medical device. To understand how we effectively kill microorganisms, we must look beyond their external shapes and examine the internal structure of the bacterial cell and the protective environments they create. Every bacterial cell is a complex living machine composed of several vital structures, including the cell wall, the cytoplasmic membrane, and the cytoplasm. The cell wall is a tough and rigid outer layer that provides the cell with its specific shape and protects it from physical damage. For the sterile processing technician, the most important feature of the cell wall is its chemical composition, which determines how the bacteria will react to a gram stain. Gram-positive bacteria possess a thick and heavy cell wall that absorbs dyes differently than the thin wall of gram-negative bacteria. During the gram stain process, gram-positive bacteria retain the primary dye and appear purple or blue under a microscope. Gram-negative bacteria have a more complex outer membrane, and after being counterstained, they appear red or pink. Recognizing these color codes is essential for identifying pathogens and selecting the appropriate chemical disinfectants. Just inside the cell wall lies the cytoplasmic membrane, which acts as a sophisticated and selective gateway for the microorganism. This thin and delicate layer is semi-permeable, meaning it regulates the flow of nutrients into the cell and the removal of metabolic waste products. The cytoplasmic membrane is also a primary site of metabolic activity where the cell generates energy. Many professional disinfectants used in our department work by attacking and disrupting this membrane, causing the internal contents of the cell to leak out, which results in immediate cell death. The interior of the cell is filled with a thick jelly-like substance known as the cytoplasm, which is roughly 80% water and contains the genetic material or DNA of the organism. This genetic blueprint is what allows the bacteria to reproduce and create exact copies of itself. One of the most significant challenges in the decontamination process is the formation of biofilm. Biofilm is not just a simple layer of bacteria but a complex and highly organized colony of microorganisms that attaches to the surface of a medical instrument. When bacteria settle on a surface, they begin to secrete a sticky and viscous substance often called a slime layer or extracellular polymeric substance, EPS. We must visualize this slime layer as a protective biological shield or armor that encases the entire colony. This matrix protects the resident bacteria from desiccation and creates a physical barrier that prevents cleaning chemicals and disinfectants from reaching the pathogens. Scientific research has shown that bacteria living within a biofilm can be up to 1,000 times more resistant to antimicrobials than the same bacteria in a free-floating state. For the sterile processing technician, the presence of biofilm means that standard rinsing or soaking is often insufficient. If an instrument is not wiped and flushed at the point of use, the bacteria have time to anchor themselves and begin building this protective matrix. Once a biofilm has matured, it becomes exceptionally difficult to remove and can lead to persistent contamination of surgical sets. We must also be vigilant for multidrug-resistant organisms, or MDROs, which are pathogens that have evolved to resist common medical treatments. Examples like methicillin-resistant Staphylococcus aureus known as MRSA and vancomycin-resistant Enterococcus known as VRE are frequently encountered in the healthcare setting. These resistant organisms are often part of the bioburden found on soiled instruments. By mastering the science of cell structures and the defensive nature of biofilms, we recognize that meticulous mechanical cleaning is the only way to physically break through these microscopic shields and ensure the safety of our patients. The structural defenses of the cell and the formation of biofilms are fueled by specific environmental conditions that allow microorganisms to thrive within the healthcare setting. To survive and colonize medical devices, bacteria require 6 fundamental environmental factors which include nutrients, water, a specific temperature range, a specific pH level, light, and the appropriate oxygen concentration. The most basic requirement is nutrients, which are provided by the organic matter left on surgical instruments such as blood, tissue, and bone. This organic soil serves as a rich food source for pathogens, which is why thorough cleaning is the most critical step in the entire processing cycle. Water is another absolute necessity for life because it is required to transport nutrients into the cell and move metabolic waste products out. Without moisture, most bacterial growth stops, although some organisms can remain dormant for long periods until water becomes available again. Temperature is perhaps the most significant environmental factor for the sterile processing technician because it determines which types of bacteria can grow in different areas of the facility. Microorganisms are classified into 3 primary groups based on their preferred temperature ranges for growth. The first group is the psychrophiles, which prefer cold temperatures between 59 degrees and 68 degrees Fahrenheit or 15 to 20 degrees Celsius. The second and most dangerous group for our profession is the mesophiles, which thrive at moderate temperatures between 68 degrees and 113 degrees Fahrenheit or 20 to 45 degrees Celsius. Because human body temperature falls exactly within this range, most pathogenic bacteria that cause healthcare-associated infections are mesophiles. The third group is the thermophiles, which prefer high temperatures between 122 degrees and 158 degrees Fahrenheit or 50 to 70 degrees Celsius. By maintaining our decontamination areas at specific low temperatures, such as 60 to 65 degrees Fahrenheit, we are actively working to inhibit the growth of these dangerous mesophilic bacteria. The chemical environment of the cell is also defined by the pH level, which measures the acidity or alkalinity of a substance on a scale from 0 to 14. Most pathogenic bacteria prefer a neutral or slightly alkaline environment with a pH level between 7 and 7.8. If the environment becomes too acidic or too alkaline, the proteins within the cell will denature and the microorganism will die. This biological fact is the reason why many of the detergents and disinfectants we use are formulated with specific pH levels to disrupt the life processes of bacteria. Additionally, light can influence microbial survival, with many bacteria preferring dark environments. Direct sunlight or ultraviolet, UV, light is often lethal to bacteria because it causes mutations in their genetic material which prevents them from functioning correctly. Oxygen requirements provide another way to classify bacteria into 3 distinct categories known as aerobic, anaerobic, and facultative. Aerobic bacteria require the presence of free oxygen to live and grow, while anaerobic bacteria can only survive in environments where oxygen is absent. A unique and highly resilient category is the facultative bacteria, which have the ability to adapt and grow in either the presence or the absence of oxygen. When these environmental needs are met, bacteria reproduce through a rapid and efficient process called binary fission. To understand this process, we must visualize a single bacterial cell beginning to grow in size as it prepares to multiply. Inside the cytoplasm, the single circular chromosome, which contains all the genetic instructions for the organism, makes an identical copy of itself. As the cell continues to elongate and stretch, these 2 DNA strands are pulled toward opposite ends of the cell. A new cell wall known as a cross wall begins to develop and grow inward from the sides across the center of the organism. This wall effectively partitions the internal contents into 2 distinct sections. Finally, the parent cell pinches in 2 at the center and separates completely to form 2 identical daughter cells. Under ideal conditions, this cycle can occur as rapidly as every 20 minutes. We must understand the clinical danger of this exponential growth because a single bacterium can multiply into more than 1 billion organisms in less than 10 hours. This is why contaminated instruments must be processed as quickly as possible to prevent the buildup of a massive bioburden. By controlling the environment and breaking this chain of reproduction, we ensure that the medical devices delivered to the operating room are safe for patient use. The rapid exponential growth of bacteria makes them a formidable threat, but to defeat these pathogens we must first be able to identify and classify them according to their physical and chemical characteristics. One of the most important and frequently used methods for bacterial identification in the clinical laboratory is the Gram stain, which was developed in 1884 by the Danish bacteriologist Hans Christian Gram. This specialized staining process allows scientists to divide almost all bacteria into two primary groups based on the structural differences in their cell walls known as gram-positive and gram-negative. For the sterile processing technician, understanding these classifications is essential because the structure of the cell wall directly determines how a microorganism will respond to chemical disinfectants and sterilization processes. We must visualize the Gram stain as a four-step chemical test that acts as a diagnostic window into the microscopic world of pathogens. The Gram stain process begins by applying a primary stain called crystal violet, which turns all bacterial cells a deep purple color. This is followed by the application of iodine, which acts as a mordant to fix the dye within the cell structure. The third and most critical step is the use of an alcohol or acetone decolorizer. In bacteria with thick cell walls, the purple dye is trapped inside, while in bacteria with thin cell walls, the dye is washed away entirely. Finally, a red counterstain called safranine is applied to the slide. When the staining process is complete, the colors provide the most intuitive way to distinguish the physical structures of the bacteria. Gram-positive bacteria will retain the primary stain and appear purple or blue under the microscope. Gram-negative bacteria which have lost the purple dye will take on the counterstain and appear red or pink. These specific colors are critical technical indicators used in the clinical setting to determine the nature of a patient's infection. Gram-positive bacteria possess a thick and multilayered cell wall composed of peptidoglycan, which acts like a heavy biological sponge that absorbs and retains the purple crystal violet dye. Common examples of gram-positive pathogens encountered in sterile processing include Staphylococcus aureus and various Streptococcus species. Many of these organisms are responsible for skin infections and pneumonia, and they are frequently found as bioburden on surgical instruments. While their thick cell wall provides physical protection, it also makes them highly susceptible to certain types of disinfectants that can easily penetrate the peptidoglycan layer. However, we must remember that some gram-positive bacilli are capable of forming endospores, which represent the highest level of microbial resistance. Gram-negative bacteria represent a more complex technical challenge in the decontamination area because they possess a much thinner peptidoglycan layer that is protected by an additional outer membrane known as the lipopolysaccharide (LPS) layer. This outer membrane acts as a selective chemical shield that can block many common disinfectants and antibiotics from reaching the interior of the cell. During the Gram stain process, this outer membrane is dissolved by the alcohol, allowing the purple dye to escape and the cells to be colored red by the counterstain. Common gram-negative pathogens include Escherichia coli and Pseudomonas aeruginosa. These organisms are notorious for their ability to survive in moist environments such as the internal channels of flexible endoscopes and the reservoirs of automated washers. Because of their protective outer membrane, gram-negative bacteria often require longer contact times with high-level disinfectants to ensure total inactivation. By mastering the differences between gram-positive purple bacteria and gram-negative red bacteria, technicians can better understand why specific cleaning and disinfection protocols must be followed with absolute precision. While the differences between gram-positive and gram-negative bacteria are significant, they represent only the beginning of the biological challenges faced in the decontamination area. Beyond normal cellular structures, some bacteria possess the extraordinary ability to transform into a highly resistant state known as an endospore or simply a spore. We must visualize a spore not as a method of reproduction but as a survival bunker for the microorganism. When a vegetative or actively growing bacterium encounters extreme environmental stress such as lack of nutrients, high heat, or exposure to chemicals, it begins to build a thick and heavy protein coat around its genetic material. This thick wall acts as a biological shield that protects the cell from the harsh conditions of the external world. A critical example of a spore-forming pathogen is Clostridium difficile or C. diff, which is a major cause of healthcare-associated diarrhea. Because C. diff forms spores, it can survive on environmental surfaces for many months and is resistant to many common disinfectants, which is why specialized sporicidal agents must be used. The existence of spores is the primary reason why high-level disinfection and sterilization processes are so rigorous. Because spores can remain dormant for many years without water or nutrients, they represent the ultimate test for our equipment. In the sterile processing department, we use this extreme resistance to our advantage by utilizing Geobacillus stereothermophilus spores as biological indicators. These specific spores are the most difficult to kill with steam sterilization, and if our process can successfully destroy them, we can be confident that all other pathogenic bacteria have also been eliminated. We must understand that a bacterium in its vegetative state is relatively easy to kill, but once it converts into a spore, it becomes 1,000 times more resistant to heat and chemicals. This biological resilience is why we must never take shortcuts in our sterilization cycles. Even more challenging than the bacterial spore is a unique category of infectious agents known as prion. Unlike bacteria or viruses, prion are not living organisms and they do not contain any genetic material like DNA or RNA. Instead, prion are abnormal or misfolded proteins that have the ability to transmit their misfolded shape onto normal proteins in the brain. These agents are responsible for transmissible spongiform encephalopathies such as Creutzfeldt-Jakob disease, CJD, which is a fatal neurodegenerative condition. Prion are primarily found in high concentrations within the central nervous system tissue, including the brain, spinal cord, and the back of the eyes. Because they are not alive, prion cannot be killed in the traditional sense, and they exhibit extreme resistance to standard sterilization temperatures and common chemical disinfectants. When instruments are used in a surgical procedure involving suspected prion tissue, specialized processing protocols must be strictly followed according to national standards and facility policy. Because prion have an affinity for metal surfaces, they can bind tightly to surgical instruments, making them exceptionally difficult to remove through mechanical cleaning alone. Standard sterilization cycles are often insufficient to deactivate prion, which is why specialized protocols such as extended exposure times in a steam sterilizer or specific chemical treatments like sodium hydroxide are often required. As sterile processing technicians, we must treat any case involving the central nervous system with the highest level of caution. By understanding the unique properties of spores and prion, we recognize that our work is the final barrier protecting the next patient from these nearly indestructible biological threats. Beyond the extreme resistance of spores and prion, we must also understand the unique biological properties of viruses, fungi, and protozoa which are frequently present in the clinical environment. Viruses are recognized as the smallest of all microorganisms, and they differ fundamentally from bacteria because they are not complete cells. Instead, a virus is simply a core of genetic material, either DNA or RNA, surrounded by a protective protein coat called a capsid. We must visualize viruses as obligate intracellular parasites, meaning they have no metabolism of their own and cannot reproduce unless they invade a living host cell and hijack its machinery to create new viral particles. In the sterile processing department, we classify viruses into two primary groups based on their external structure, which are enveloped and non-enveloped viruses. Enveloped viruses possess an additional outer layer composed of lipids or fats which they steal from the host cell membrane. While this envelope helps the virus attach to new cells, it also makes them highly susceptible to heat and common chemical disinfectants because the lipid layer is easily dissolved by low-level disinfectants or alcohol. Common examples of enveloped viruses include hepatitis B, hepatitis C, and HIV. Non-enveloped viruses lack this fatty layer and are composed only of tough proteins, making them much more resistant to environmental stress and chemical inactivation. Examples such as norovirus and rhinovirus require more rigorous disinfection protocols and longer contact times to ensure they are fully destroyed. By understanding this structural difference, technicians can appreciate why standard precautions and specific contact times for disinfectants are mandatory for every instrument tray. Fungi are a diverse group of microorganisms that include single-celled yeasts and multi-celled molds. Unlike bacteria, fungi are typically decomposers that thrive in dark, moist environments, and many species are aerobic, meaning they require oxygen to survive. Molds reproduce by releasing tiny spores into the air, which can travel to new areas and begin to grow. It is critical for the technician to distinguish between fungal spores and bacterial endospores. While fungal spores are used for reproduction, bacterial endospores are used solely for survival in harsh conditions. In the healthcare setting, fungi are primarily considered opportunistic pathogens, meaning they cause infection in patients with weakened immune systems or through contaminated medical devices. Because fungi can produce persistent environmental bioburden, the thorough cleaning and drying of instruments is essential to prevent fungal colonization. Protozoa are the most complex group of single-celled microorganisms and are often classified as tiny animals due to their ability to move and hunt for food. They are typically found in water and moist soil, and they require significant moisture to remain active. Some protozoa can form protective cysts, which are thick-walled structures that allow them to survive in harsh conditions outside of a host for extended periods. While protozoa are less commonly linked to surgical site infections compared to bacteria, they represent a significant biological hazard in the processing of equipment that comes into contact with contaminated water or mucous membranes. By recognizing the diverse properties of viruses, fungi, and protozoa, sterile processing technicians ensure that their decontamination practices are comprehensive enough to neutralize every type of microscopic threat. To conclude our comprehensive study of microbiology, we must reflect on how this technical knowledge transforms the daily tasks of the sterile processing technician into a critical clinical science. By mastering the concepts of the micron, the morphological shapes of bacteria, and the complex structures of viruses and prion, we recognize that our work is the first line of defense against healthcare-associated infections. Every instrument we process represents a biological battleground where we must use our knowledge to defeat invisible pathogens. We understand that the microscopic size of bacteria, measuring only 1 to 2 microns, means that even the smallest amount of organic soil can harbor thousands of dangerous microorganisms. This is why we must apply total technical precision to every task, from the initial point-of-use treatment to the final stages of sterilization. The knowledge of microbial resistance hierarchies dictates how we approach our duties in the decontamination area. We recognize that while vegetative bacteria like Staphylococcus aureus are relatively easy to kill, specialized threats like Geobacillus stereothermophilus spores and infectious prion require much more rigorous processing. This understanding reinforces our surgical conscience, which is the inner drive to do what is right even when no one is watching. When we encounter multidrug-resistant organisms, MDROs, like MRSA, or resilient bioburden like C. diff, we do not take shortcuts because we know the biological reality of these threats. We understand that if we fail to remove the sticky slime layer of a biofilm, our chemical disinfectants will be unable to reach and destroy the pathogens hiding beneath the surface. Our professional responsibility also extends to the strict adherence to the manufacturer's instructions for use, or IFU. Because different microorganisms exhibit varying levels of resistance, the specific temperatures, contact times, and chemical concentrations listed in the IFU are not suggestions but are mandatory requirements based on biological science. By understanding microbiology, we appreciate why a specific contact time for a disinfectant is necessary to penetrate the fatty envelope of a virus or the thick cell wall of a gram-positive bacterium. We recognize that our technical expertise and our ethical commitment are the only things standing between a sterile instrument and a contaminated device. This mastery of microbiology ensures that every surgical set delivered to the operating room is safe, functional, and free of all viable microorganisms. By breaking the cycle of microbial survival, we fulfill our primary mission of protecting the patient and maintaining the highest standards of professional excellence in the medical center. This concludes our study of Chapter 4.